Hello, welcome back to the fourth week of the Jerusalem Science Contest. Your exam this week will be on chapters 10 and 11. That's animal systems and animal reproduction. And I've chosen as my topic today something that I've taken from chapter 11, which has to do with cloning. So here you see a beautiful young girl, and what I've done is, it's actually my granddaughter Zoe, and I've cloned her. Should have gotten permission, I guess, first from her father, but I've made 5 copies of her here, so we now have 6 of her, which would be great for me because my wife and I don't get to see her very often because she lives out in California. Anyway, cloning is nothing more than making multiple copies of an animal, or in this case, a human being. Of course, we don't clone human beings, but I just use this to illustrate that, in fact, if it were possible to do that, we could make these copies of a person as well. And of course, there are all kinds of problems associated with even trying to do something like that. First of all, it would be illegal, for one thing. And secondly, there are a bunch of moral, ethical issues that would be involved when we talk about human cloning. And some of the same issues actually occur when we're talking about animal cloning, but I think the problems are less severe. So the first thing I'd like to talk about is the idea of genomic imprinting. And the question might be, are identical twins and clones alike? And what I'm telling you here is that they're not exactly alike. If you've ever seen so-called identical twins, is that they're not really identical. And here are actually 2 animals. The one on the right, the cow on the right, is I believe was the parent animal, and this cow is the clone of that animal. And as you can see, although the animals look very, very similar, if you look at the markings, you can see that there's distinct differences in the marking, and that has to do with what is known as genomic imprinting. And basically, this involves a process known as epigenetic reprogramming. So this, to illustrate that, I've taken this particular kind of mouse, which is called an agouti mouse, and the agouti gene, this particular gene, we have on the gene upstream from here. There's a region of the gene that's known as the variable methylation region, and you can have mice in which this region is methylated. Remember, we talked about epigenetics, I believe it was last week or perhaps the week before, when we spoke about epigenetics, and we talked about methylation as one of the mechanisms that actually can change the epigenome. So here you have basically a normal mouse, and this variable methylation region is completely methylated. Now, it's possible to demethylate that. So you have the non-methylated region here, and then you get the agouti mouse. Now, the first thing that you notice about the 2 mice is that this one is a normal gray or brown mouse, and this one has a yellow coat to it. This mouse is healthy. This mouse, because of other changes that have occurred during this demethylation process, the mouse will ultimately become obese, and it will probably develop diabetes during adulthood. So these are some of the problems that are associated with trying to change the epigenome through this reprogramming process. Now, epigenetic reprogramming events can be either of the gametogenic nature, which means that what we've done is we've taken the actual gametes, it could be the sperm or the egg, and we've done the reprogramming at that particular point. Now, since these are germ cells, what that means is it's going to affect future generations. So once you've reprogrammed these things, then the future generations are going to have that same reprogramming. And the other possibility is to use what is known as pre-implantation reprogramming. Here we've already got a zygote. We've already got could be a morula or a blastula. Some cells, the egg has already been fertilized, and these cells are now beginning to divide. And at this point, reprogramming could occur. And most of the cloning defects that actually do occur seem to occur during this kind of pre-implantation reprogramming procedures. So that's a problem associated with using that particular type of reprogramming. Now here, something very much like this is probably in your book, but just showing you how if you were going to clone a sheep, what you might do is you could take a cell from the a mammary cell from the udder of the sheep. So we're taking it from this sheep here, and then we removed DNA. We've actually removed the nucleus from an unfertilized egg from the second sheep here. Now we take the two denucleated udder cell here, and we take the nucleus from this cell, and we put both of them together. And then this is usually hit with a small electrical current, which actually causes fusion. And then we're going to culture the cell for a period of time, just to make sure that the cell is actually viable, that it's alive, and it's going to reproduce. And then it's implanted into a surrogate animal. So this is an animal that's been given certain drugs to ensure that it is ready for implantation. And ultimately, what will happen is when this animal gives birth, it will actually not give birth to an animal that has its genome, but it will be an animal that will have the genome of the original mammary cell donor, which is the sheep shown up here. So this shows you a photomicrograph of the actual cloning process of an egg. So here you see the egg. This pipette here is holding the egg in place, and this very, very fine, it's actually much smaller diameter than a human hair pipette, specially drawn out pipette, going to be inserted into the egg. And either, I don't know, I can't tell from this picture whether this is an egg that hasn't been denucleated first. So the nucleus will be withdrawn from the egg, and then you can use a similar kind of pipette and actually inject the new nucleus into the egg. And this is all done using stereotactic equipment. It's kind of like if you looked at a microscope stage that actually had all this stuff mounted on it, and you could very, very carefully manipulate the needle very slowly, moving it over towards the egg. I've actually done this about 5 years ago or 6 years ago. I was at the Weizmann Institute in Rehovot, and I learned this technique and actually applied it to not to cloning per se, but it was a similar kind of technique where we were inserting GFP, which is called green fluorescent protein, or the RNA that is responsible for making that, into the egg of a zebrafish, and that would produce a zebrafish, the cells of which would actually start making this green fluorescent protein. But it's a similar kind of technique where you have to come in very, very carefully, and you want to be, in this particular case, we had to make sure that we actually got into the nucleus, so you have to get between the cell membrane here and then the nucleus, and if you inject at the wrong point, nothing's going to happen. So you have to very, very carefully manipulate this pipette. But that's a technique that can be learned and can be done with great success. And first sheep that was actually cloned was a very famous sheep named Dolly. Although you may not be aware of it, that was in 1996. Dolly also had several other clone mates. So from that same donor sheep, there were 6 other sheep that were produced, and these are Dolly's clones. Well, they're not clones of Dolly, but they're clones of that original animal. So all of these animals are these animals, and Dolly and also the original animal would be genetically identical. And here I show you some other clones. This picture we've already seen. Two cattle. This was the parent animal, and this is its clone. Here you see again I guess they're cattle here, two types of cattle. And the larger one is, both of these are actually clones. It's just that this is an older one and this is the smaller one here. Here are 4 or 5 piglets that were cloned. And this last one, you notice that these two animals are not the same, but this animal here, this cow was actually cloned, and this is this cow's healthy offspring. So this offspring is not the clone of this animal, but was produced naturally through just the regular fertilization process. So that's just demonstrating that these creatures can actually reproduce, and they can have a healthy offspring, although that doesn't always happen. Now here is kind of a financial reason why somebody might want to clone an animal. I don't remember the name of this animal, but this was a famous bull which was in great demand, and it was determined that this animal, if it were cloned, would actually be something that would be very, very profitable to the individual that was doing the cloning. And it turned out it takes something like cost maybe $50,000 to complete the cloning process. However, this animal was sold for $170,000, so that's still a substantial gain for the individual that owned the animal. Now, this is an interesting kind of thing that you could do. So this animal, which is called a bucardo, became extinct at the beginning of this century. So that was only 14 or maybe 15 years ago. The last bucardo died out, but some of its tissue was preserved. And once the technique was developed, a bucardo was actually cloned, and it was implanted into, I guess this is some kind of a goat or sheep, I'm not even sure what it's close to, but it was implanted into a living animal that was not of exactly the same species, but nevertheless that animal carried this to term. And when the bucardo was born, it didn't survive very long. It was born live, but it rapidly succumbed to a series of infections. So this process really needs to be improved before we can actually successfully bring back an animal that has become extinct. Of course, there's a lot of questions about whether that should actually be done or not. Perhaps when an animal becomes extinct, if it's not man that caused the extinction, such as through overhunting, etc., but if it's a natural type of process, perhaps it's best that we let nature take its course and not bring back an animal from extinction. This is something that I need to at least discuss, although as I mentioned before, we would not really, we're at the point right now where there's a lot of debate concerning human cloning. And by human cloning, I'm not really talking about trying to make a human being, taking a person and then cloning that individual and growing an entirely new individual. I don't think anybody right now wants to see that happen. But there are people that would be interested in taking, just for reproductive purposes, people that would be unable to actually have a child. One of the two parents actually has genetic material that could be provided in this particular case. It might be possible to produce a viable clone. But I don't think anybody's actually doing anything like that. But that would be one possible thing that you could do for reproductive purposes. The other one, I think, which is much more probable, would have to do with stem cell production. So, and even this has a lot of problems associated with it, not just the problems of actually carrying out the process, but problems associated with the ethical and religious implications of doing this kind of thing. But initially, with stem cell production, embryonic stem cells are really the best stem cells that one could use. If one wanted to clone those, that would be the best type of cell that you could use, because it's the least differentiated type of cell. So if one wanted to grow tissue, Mice, for instance, that embryonic stem cells were taken from a mouse. There are strains of mice that actually have Parkinson's disease, and these Parkinson's mice, when they were injected with stem cells that had been, let's say, reprogrammed, cells themselves would actually start to proliferate, and then the symptoms of Parkinson's disease in those mice would be alleviated. So if this could be applied to human beings, this would be a way of actually treating things like Parkinson's. So that's one of the reasons why people would like to be able to do it, and ultimately it might be possible to actually replace entire organ systems, or to regenerate a limb that had been lost through some kind of an accident. So this slide is just showing you the entire continuum of reproductive technologies, all the way from natural service, which is just selecting two animals and allowing those two animals to mate in the natural way. So this would be natural service. And then an animal could also be artificially inseminated. So we'd have a female, and then the sperm from a donor animal, and then you artificially inseminate that animal. Another possibility is embryo transfer. In embryo transfer, what you would be doing is just removing the embryo from one animal and replacing it into a second animal. And there might be a number of reasons why you might want to do that. If it turned out that the host animal, or the original mother, became very, very ill, and the embryo was about to die, then it would be possible to take that embryo out and put it into another animal that had been prepared for the transfer. The other one is in vitro fertilization, where we actually could take the eggs, the ova of one animal, put them in a petri dish, and add the sperm of another animal, and then watch as these cells divided, and then reimplant one or more of the embryos into a host animal. Another one that is used sometimes, but it's not extremely simple to do, is known as embryo splitting. And this again involves taking something that's very, very small. You could have a group of, let's say, 16 cells, and actually splitting them in half so that you have 8 cells and another 8 cells. And then each one of those, this is essentially making a twin of an animal. This is basically what happens naturally. An embryo splits, and each of the subdivisions grows, and that's how you get identical twins. But the last two are the ones that we'd be most concerned with for purposes of this talk, and that's blastomere nuclear transfer and somatic cell nuclear transfer. So blastomere nuclear transfer is what I was describing when I talked about the way you'd make a sheet, the way Dolly was made. I'm sorry, that really is somatic cell nuclear transfer. So the somatic cell was the cell that was taken from the udder, or from the mammary gland, and the transfer was done that way. In blastomere nuclear transfer, so I misspoke, blastomere nuclear transfer would be actually removing nuclei once you already had a fertilized egg that had begun to divide, to remove the nucleus of that and then transfer the nucleus of another animal into that, and again isolate those cells, culture those cells before they're re-implanted into a host animal. All right, so what are the problems that are associated with cloning? Well, if you're cloning pigs and goats, and for those of us that observe kashrut, we're not interested in pigs at all. And for those of you that have never eaten goat, I actually found at our one of our kosher butcher shops, they were selling goat not that long ago, and actually tried goat. Did not like it particularly. I don't think I'd ever want to eat it again. But I had something that I guess you would call a goat chop, which looked exactly like a lamb chop. The taste was somewhat similar, but it was a little off-tasting to me, so I don't think I'd want to eat it again. But nevertheless, there are a lot of people out there that do eat goats, and there are many other people that eat pigs. And so if you clone pigs and goats, you find that the clones have low mortality and morbidity rates. So they basically, the offspring from these clonings don't usually die. I mean, they usually have normal or relatively normal lifespans, and they don't become sick. That's not true of cattle and sheep. There's a lot of problems with cattle and sheep. So one of them is called LOS, which is large offspring syndrome. And what is characterized in this syndrome is an abnormal development of the placenta. And that seems to lead to these other two conditions. Hydroallantois is actually a condition in which there's extra amniotic fluid. And that causes, because of that, there's kind of an increased pressure inside the amnion, and that can cause some problems in the development of the embryo. And the embryo tends to be very large, and because of that, you have dystocia, which is actually a difficulty in the cow delivering this large size offspring when it comes to full term. So a lot of times that would mean that the cow is going to have to be surgically delivered, which nobody wants to do. Or if you try to deliver if the offspring is attempted to be delivered naturally, both the cow and the calf might die. Other problems that are associated with both cattle and sheep are respiratory problems, prolonged recumbency, which means that basically the mother, and again I think this probably has to do a lot with the fact that she's carrying this enlarged fetus, or has been carrying it for a long period of time, is that she remains laying down for a very, very long period of time because she doesn't get up. She develops something that's kind of like having bed sores, and there are other problems associated with that. She'll develop muscle weakness, and there are other difficulties that she's ultimately going to have later on in trying to walk and to move around. There's an enlarged umbilical cord. The offspring may have hyper or hypothermia, that is difficulty in regulating its body temperature. Contracted flexor tendons, which means that the animal is going to have probably trouble walking. Again, that is not the parent, but the offspring will have trouble. And also abnormal development of major organs, which is one of the reasons why that only about, probably right now, maybe 20% of sheep and cattle that are actually produced by this process are viable, have long-term viability. That is, that they actually remain healthy and will have a normal lifespan. What are the benefits of using this particular technique? Well, for one thing, if you're trying to raise an animal, if it's a dairy cow, you're raising it for its milk. If it's a beef cattle, they're being raised because their meat is valuable. Sheep are valuable again for meat, and I guess some people might even use sheep milk as well. And also they're producing wool. And some animals are identified that have exceptionally good meat or good milk, or they produce very, very good wool. So if you actually were to clone those animals, that means that the products of those animals would be consistently of high quality. The second thing is the fact that animal health can be improved in this fashion, because you can select animals for certain traits. Once you have an animal that doesn't have any known genetic problems, you can actually get a strain of animals that are going to be just naturally will become healthier and will have greater vigor. Reduction in the number of unwanted animals. It turns out that in this particular technique, when I say an unwanted animal, for instance, if people are raising dairy cattle, they don't want males, they want females. Males obviously can't produce milk. The male can only be used actually for reproductive purposes, but you only need, one male could service many, many females, so you don't need very many males. What happens to the dairy cattle males, the unwanted ones that are born, is that they're raised as veal. So they're put in these kind of very unhealthy types of conditions where the animal becomes anemic. It doesn't move around a lot, and even though people eat veal, veal is really it's the way it's being produced. It's probably not really very, very kind to the animal that is being used. So in this way, you can reduce the number of unwanted animals, because all you're going to do is clone the female animals, and you're not going to get these additional males, these excess males that you don't want. Environmental advantages in moving from a grain-fed to a grass-fed economy become obvious, because grain is very, very expensive. Now, it turns out that grain-fed animals tend to be a lot tastier if you're going to be eating them than grass-fed animals. But every now and then, it turns out that some of the grass-fed animals actually are comparable in quality to the grain-fed animals. So if you have these grass-fed animals that you've identified, a group of them, then if you clone those animals, you'll be getting be moving from a grass-fed to a grain-fed type of economy. So that means there's going to also be potential savings to the consumer, and the ranchers are going to benefit as well. Since it doesn't cost as much money to raise an animal, because you don't have to pay for all the feed, and you're just going to let these things roam around and eat the grass, free-ranging animals. The ranchers are also going to benefit financially from that as well, and they'll have healthier and higher quality livestock. There's a lot of discussion that still needs to be done regarding cloning, but as the procedures get better and better, I think a lot of the problems that we currently have with cloning, but the safety concerns, the concerns that some people have expressed regarding the quality of meat that would come from cloning, really do not warrant any real concern, because these things have been reviewed. All the data has been reviewed by the Food and Drug Administration, and it's been concluded that animals that have been cloned, their meat would be no more harmful than the meat of any other animal. So anyway, again, I wish you good luck on the exam, and we'll start next week on chapters 12 and 13. Chapter 12 is animal growth, and chapter 13 is plant and animal diseases. And I think my lecture is coming from, I believe, from that chapter. I'll be doing plant and animal diseases and weed science simultaneously. I think that may be the case. So we may be skipping one of those chapters, but I will let you know before the next lecture. Again, thank you for your attention.